Well, hello and good day, everyone. Thank you so much for joining us in this webinar today that we are going to be talking about breast cancer risk, diving into looking at the genetics that lead to that or could possibly lead to that. We're going to be diving in and looking at how the body metabolizes estrogen. How do we detoxify estrogen? Looking at different ways of possible hormonal imbalances and different ways that we can help to have our estrogen, our hormones balanced, and how that looks. I am so honored to be joined by one of my dear friends and colleagues, Dr. Stephanie Nielsen. Hello and good evening to you. How are you?
I'm well. How about you?
I am doing fantastic. I always love to be here with my colleagues and with our audience because they're always so eager and ready to learn. And just we have such brilliant, awesome people who come to these webinars, and I really appreciate every single one of them. Actually, I would love to see where you're coming from. Pop in the chat where in the world you are right now. Make sure our chat is working for everyone. Also, please take a moment, fill out the poll questions. They really help us to focus our energies and the information that we provide to you to make sure you are getting out of this time that you want. Wonderful. Look at this. So we've got Miami, or let's see, in California, Oregon, Croatia. Wow, way across the world. St. Louis, Toronto, Indianapolis, Canada, Texas,
Nina
New
comes frequently
Hampshire.
from Croatia. In fact, we all have invitations over to her apartments. She's got 3 apartments and we have invitations to them. So
Oh,
we love
beautiful.
seeing Nina.
Yes, so glad you've joined us.
Oh my goodness, coast to coast, from Miami to
Absolutely.
the Bay Area. I love it. And Canada, Quebec, Texas, New Hampshire, Tennessee. We have women coast to coast that are interested in taking care of their health. And Wisconsin, I was born in Wisconsin. How about that? Love it.
So yes, please make sure you fill out the poll, and thank you if you already have. We really want to make sure that we are answering the questions you want here. So making this ROI, your return on investment, worth it. So, all right. So let's dive into estrogen. What about it? About the metabolization and it's like estrogen itself. I don't know how many of you realize that there are actually several forms of estrogen that we have, and each one works a little differently. And the breakdowns and into the metabolism of them and those metabolites and then how they work in our system. And I'm getting a little ahead of myself. So, Dr. Stephanie, what would you like to share first and foremost on estrogen?
Well, I want to look at our poll questions. I want to see what our clients want to look at, and then I want to jump
Absolutely.
into their questions. So, powers that be, if we've got the poll, there we go. What brings you? I have a family history, 30%, and that is scary because you may know the statistic. One in every 8 women will be touched by breast cancer at some time in their life. And while breast cancer is only 1 to 8% of the population, there is more and more incidents, not necessarily genetic, though we do want to discuss the genetics tonight, but your inability to detox. So detox is going to be part of what we talk about. We've got some healthcare providers. Welcome. We always love healthcare providers. You're interested in genetic testing. Great. That means you may not have had genetic testing done yet for your hormones. Hey, we have 16% that are breast cancer survivors. Welcome. We're so glad you're here. And you know what? We'd love to have you pipe in tonight. There may be things that you learned along your journey that you'd like to share. And then we've got about 20% that says we're curious. And curious makes sense since so many lives are tested and touched by friends or family or others. So are you currently aware of estrogen detox profile? Yes, I am. I've done my genetic testing. We have 34%. I think I know, but I'm not 100%. No, but I'd like to know. And what is estrogen detoxification? Okay, good to know. So we have several people here who are quasi-educated, Dr. Varden. And so one of the things that I love is in our DNA 360 test, we have a magnificent red stoplight, green stoplight, yellow stoplight example. And you were so instrumental in making sure that that works properly. We'll have to go through that. If you do have your test, pull it up so that when she goes through that, you'll get to follow it. If you don't have your test, don't worry. We might be able to share one on profile. In fact, Katie, if you have a test example of a test we can show that when Dr. Varden goes through estrogen toxicity, it'd be great if we could show them those stoplights. Have you ever had your hormones tested? 12% says saliva, 32 said blood or serum, 16% urine. There were no others. We were just trying to discuss what other others there could be. There are some loosely second or third generation correlations, but not really testing. And then no, 40% have not have tested. This, okay, so I'm going to have a hot button about getting tested because we want to test, not guess. Whether it's your genetic testing or whether it's your blood or serum testing, you don't know what you don't know. And we don't want to say, oh, I should be on a hormone phytoestrogen enhancer. I need to be on DIM to decrease it. I need to be, how do you know? If you're not testing, you're guessing. And the biggest waste of money you can have is when if you don't know something's doing what it's doing for you. Which area are you most interested today? Oh, and Lara, you hit it right above. Genetics of breast cancer, 20%. Estrogen detox. Here we go. It's going to be an estrogen detox night, girls. Hold on to your horses. 42% lifestyle and nutrition, 34%, hormone balancing, 32%, and then all of the above. 70% said, lay it on me. So I think we're going to do a lot of estrogen detox, some suggestions on how we can balance those out and try to wrap it all up with a bow.
Well, I'll tell you, we've got our work cut out for us.
Uh-huh.
You know, it's really interesting that, when we're talking about different statistics for breast cancer, they actually have found that 85% of breast cancer cases occur in women with no family history of the disease. I mean, we just saw that, 35%, I think it was, or so, had family members with it. But even looking now, we are finding that breast cancer is occurring in women at a younger age, as well as those that don't have significant family history of it. And that is really important. And I mean, profound for us to realize, each of us as individuals, as women, but as practitioners, because I think this really emphasizes the role in our environment, in our hormones, in knowing our detoxification pathways and how best to support them, how to manipulate and really influence epigenetics, the way that our genes are expressed, so that way we can support our bodies. That it is very important to know your genes, so you know what you have to work with, your baseline. These are the cards you've been dealt. Now, how do we play that game? How do we work those cards in our favor? Because we really need to shift that focus from fatalism. Oh, I've got, the BRCA, mutations, and really shift it over to that empowerment through
So let's
what
break
we do
those
about.
down. Let's break that down for a second. According to recent literature, and we like to be literature based, 1 in every 8 women, 12.5%, are likely to have some form of breast cancer throughout their life. Okay. So That's not a ton, but it's significant. Now, many of you have probably heard of the BRCA1 or the BRCA2 mutations. Interestingly, though, more and more of breast cancer is not necessarily heredity related. So BRCA1 is probably the most significant, but only for certain populations, and it is 35% of all the heredity or hereditary breast cancer, while BRCA2 is about 25% of the hereditary breast cancers. But since we know that there's only a small portion of the population that has the hereditary, the rest are non-hereditary. I like to tell people, and Lara, I'd be interested in what you think. I like to tell people, I'd rather you plan that you could go into inflammation. You could go into mutation. So how do we clean up that chronic inflammation and environment to lower the risk of that? It hurts my heart when I see women, they find out they have BRCA and then they instantly go mutilate themselves by cutting off their chests or cutting off their breasts. If that's what the excuse me, a decision that they made, I support them in that. But I think there's a fear-based knee-jerk response because, again, only a small portion of these cancers are heredity based. And if you, let's say you do have a mastectomy or a double mastectomy, and yet your cancer was not heredity based, it was environmentally based, or your detox pathways weren't working, you're going to pop it back up. Now, I don't know if you know this, but you can get breast cancer again after you've had a mastectomy. So just because you have a gene, as we know with many of the genes, having a gene does not doom you to having a chronic disease. Lara, what would you like to add to that?
Well, it's also having people understand the BRCA genes. I mean, and I will disclaim this, that we do not run those particular markers in our DNA 360 test, because that is actually looking at the branches and the leaves. Okay, that is looking at a specific disease or associated disease state. We are looking at more the root cause, the terrain, because, and what Dr. Stephanie was saying, just because someone could have a mutation in the BRCA genes does not mean that they will get breast cancer. And this is what we're talking about is, cleaning up your environment, knowing what you have to work with to make sure that you have the prevention of not going down that pathway. But I want to make sure that we have clarity in what the BRCA genes are. These are tumor suppressor genes. Everyone has them. So when people say, oh, I have the BRCA gene, well, yeah, of course you do, but is it mutated or not? Now, tumor suppressor genes, it basically means that these normally help prevent cells from becoming cancerous. And what ends up happening is that when there is a mutation in these genes, that means that their role in repairing the DNA is not working so well. You can think of them like the DNA repairman. Okay, they help to fix broken DNA in cells. When these repairmen have a problem due to the mutation, the cells can accumulate too much DNA damage. And therefore, as they're splitting and growing, they're more likely to become cancerous. That's how that works. And I want you to hear the words, they are more likely. It doesn't mean that they are absolutely going to. This is where the lifestyle, diet, and all of these other wonderful things comes in. But also understanding and testing, not guessing, knowing that, okay, you've got your genes, but how are those genes, how are your hormones actually expressing? And this is where some of those other tests come in, which we are going to talk about shortly.
Right. So if you're gonna look at your DNA 360 test And let's go ahead and everybody who has done your DNA 360, let's go ahead and put a 1 in the chat. I just want to see if we've got 10 of you, 20 of you, 60 of you. If you've done your DNA, okay, so I'm seeing a few that are coming up. Great. And then put a 2 in if you've not done your DNA 360, because there's a lot of companies that will put in like the BRCA gene. So there's a company, it's like 45 and you, that will say you have BRCA or you don't have BRCA, but they're only testing for a small mutation there, and they're only testing for Ashkenazi Jews. Well, I'm not an Ashkenazi Jew, and so that isn't applicable. One of the great things about the DNA company is we have 73 genes that we test that are actionable. So I would say a majority of our people tonight, is that what you're seeing, Dr. Lara? A
Oh yeah,
majority
the
of them
majority
have
of time
done.
done. Yep.
Yeah. Okay. So I see a handful of 2s, but mainly 1s. So for those of you who've done your DNA 360, you know, if you go to your hormone profile, we have in there about estrogen toxicity, estrogen dominance, and then your
Hormone
detox path.
profile.
Hormone.
Yeah, because we list what your hormone profile is, whether you have a dominant profile, a balanced profile, whether it's androgen, estrogen, so that really depends on the combination of all of these players working together that provides us a genetic hormone profile. That doesn't mean that's what you are currently expressing in your hormones, especially when you consider if you are perimenopausal, menopausal, postmenopausal, your hormones are going to be, different. So again, this is based on a genetic perspective.
So remember, the genetic perspective is your blueprint. This is what your DNA says. This is likely how you will express. So it's your blueprint, just like if you've got a blueprint of a house. Then we have how you're testing. Are you testing serum? Are you testing saliva? Are you testing urine? That shows how you're actually expressing. So we've got to layer the two. I have a tendency toward estrogen dominance. I have a tendency toward estrogen toxicity. Am I expressing it? No. Okay, great, because all your levels are normal. Wonderful. We're happy to hear that. Or you do it through urine, such as a 24-hour urine collection or a Dutch test, and then we can look at the specific pathways. Now, Lara, Katie, did you find that you could put up so that everybody could see one of our tests and look at our beautiful stop sign?
We sent it to Dr. Barden's email.
Woohoo! While she pulls that up, then we're going to jump that in so that she can share that. Thank you so much, guys. That way we can go through our sex hormone pathway and show you, if you haven't done your DNA 360, what you get. And if you do, while she's pulling it up, then I'm going to recommend that you pull it out, because this is really cool. We have a step 1, step 2, step 3 to show how the hormones go through, how the toxicity can build up or not build up. And then we'll talk about each one of those steps and the genes related. Now, I love this. This to me is fun. I know a lot of people are going to go, oh my gosh, this gets a little crazy. All I'm going to tell you is absorb what you can absorb. Don't worry about what you miss. You're going to get the replay. If you don't know this, you get a replay of each of our webinars. If you're on our webinar list, you get invited to them all and you can go back and see them. If you have to leave a little bit early, it's okay. You can catch the rest and all our question and answers. But the beautiful part of this is take what you can do, go through along with us, or just sit and listen and then go back and look later when you've got your test up. Take a little bite at a time, because nobody learns this overnight. And the most important thing is how it impacts you and the little teeny steps you can do to get yourself healthier. Lara, did you find it?
Yes, I've got it up. And actually, I do have to give a little bit of a clarification on this, that we don't actually have the sex hormone pathway chart in the patient reports. It is actually found in the clinician's report. And so really it's up to the clinician to give it to the client. With my clients, you know, when I have someone come in, especially if they are focusing on or having hormones be a part of their program, I actually, you know, have this chart for them, you know, and give it to them or have it uploaded, you know, in the medical records platform. But this isn't something that you will find in your actual hormones reports or
Patient report.
So in
My
case
bad. My bad.
that's okay. It's so if you're frantically looking when you see mine,
I lied to you.
where the heck is
I
it?
lied to you.
That's okay. So the other thing is that the ones that were so generously shared via email were not the ones that are in the clinician's report either. So what I'm going to do, and I've done this before, so I've did this over a year ago when I was walking through these reports. I'm going to show you mine. So this is my personal report, and I don't mind sharing that. So let me. Okay. So do you see on my screen, do you see that all right?
I do.
Okay. Please put a yes in the chat if you can see it. I just want to make sure that everyone is viewing and that it is clear. Beautiful. Thank you so much.
I love your guys' feedback. Thank you so much.
You guys are awesome. Okay, so when we're talking about the sex hormone pathways and pathway charts, now, you know, when you are looking at your reports, you're going to see, you know, a lot of the genes and, you know, the particular genotype, the, you know, on the GST genes are, I'm sorry, on the UGT genes, on a couple of them, there's going to be a number because we're looking at copy number variants. And it does have some brief little descriptions further down the page of your sex hormones when you're looking at that report. I believe it's in the summary report.
Just a minute, Lara.
Yes.
Can somebody from tech help Mary? Oh, good. Okay. I think they are already. Perfect. Sorry.
Okay. So when I walk through this, it will make a lot more sense, those descriptors, where it tells you the gene, your genotype, and then gives you a brief little information about it. I'm more of a visual person. I much, you know, prefer this, but it just makes that to have sense. So even though you may not have your sex hormone pathway chart that I'm sharing right now, you can translate the information in your reports to visualize what's going on. So in step one, this blue row across the top, this is telling us how our androgens, our main hormone, which actually is pregnenolone, but we start at progesterone, how that works to help determine the particular hormone profile that we were mentioning a little bit earlier. So progesterone, everyone makes this, men and women. Men and women make testosterone and estrogen as well, just in different concentrations. So the progesterone gets converted to testosterone with the CYP17A1. So that's CYP, which is part of the cytochrome P450 family. This is part of the phase 1 detoxification that happens in the liver. And it's a matter of how it converts one molecule to another. So
Lara, will you circle and show them where on that chart, just in case if somebody wants to
see
follow?
this? Do you see my cursor?
Yes, we do.
Okay, so it's this first one here on the left. So progesterone converts to testosterone with the CYP17A1 gene. Now, one thing with the street lights, okay, green, yellow, red, I want to preface that when it's green, it doesn't mean green is good, red is bad. This is really strictly the rate of which the particular enzymatic reaction is happening. Okay, so it just is, and it's just how to read it.
So green is fast or faster,
correct.
Red is slow or slower. So remember, it's not good or bad. It's how fast do you get converted? How slow do you get converted? Because that's going to show us, are we going to be dominant or not dominant?
Correct. And when you see a yellow, generally that means that there will be some sort of mixture, like a moderate rate, okay? That it's kind of like that somewhere in between. So I happen to convert progesterone into testosterone at a slightly increased rate, at that moderate increased rate. So this right here, that speed determines that I have a dominant profile. Now, if this were red, if I converted progesterone to testosterone in a slow or reduced rate, I would have a balanced profile. So now we've kind of determined, if looking at yours, whether you are dominant or balanced. Now we have to figure out, is it estrogen dominant? Is it androgen dominant? Or is it co? Now, the way we figure this out is we are looking at the other pathways testosterone can take, the accumulation of how these work together. So now we have a pool full of testosterone. Testosterone can do a lot of different things. 4 of the main pathways that it takes is, one, it's cleared. It's cleared through the UGT, CYP3A4. This is the glucuronidation pathway. And I happen to clear androgens, testosterone out very quickly. So that leaves less of a pool of testosterone available for use, but I also make testosterone rather quickly. So it's this beautiful dance of where it goes and how quickly it comes and goes. So another pathway testosterone can take is it can bind to the androgen receptor. That's what the AR stands for, androgen receptor. Now, I have an increased binding affinity, meaning that when there is a testosterone receptor near, testosterone will bind very easily, very quickly, very efficiently. So when I give my body testosterone, and when it's there, I use it well. But I have to, my body gives me what it asks for. So if I'm working out, if I'm, doing things, then, I can get that usage very easily, very quickly. Another way that testosterone can take is being converted to dihydrotestosterone. Now, dihydrotestosterone is about 3 to 5 times more potent than testosterone itself. It's the very bioactive, even stronger form of testosterone. I convert very slowly testosterone to dihydrotestosterone, and actually, that's optimal for women. We don't necessarily want to, that's more like a manly man type of thing. But
well, not only that, that's how you get female pattern baldness. So if you convert quickly, you may be losing your hair. So we want to look at your estrogen toxicity and your conversion to make sure that you're somebody who can maintain a head of hair.
Yeah, I mean, we like our hair.
We do like our
I'll tell
hair.
you though, there is a way. I mean, I may convert slowly, but when my body was in a lot of trauma from a car accident, from surgeries and everything else, and being under anesthesia, just the stress of all of that made my hair thin. I mean, it was thin. I had some, a little bit of balding going on, and yeah, talk about a little ego bruise. But yet this is my genetic predisposition. So don't think just because, estrogen dominant normally, genetically speaking, would have thicker hair, not have that pattern balding, but it can still happen due to other stresses, other environmental factors, other toxins in the body. But again, this is why we want to know the genetics, the pathways, so that way we can help support that. And if you do happen to convert quickly testosterone and dihydrotestosterone, there are supplements, there are things that can be done to help alleviate that. So getting to the final pathway that testosterone can take, it can be aromatized, converted into estrogen. And yes, testosterone turns into estrogen through the CYP19A1 gene. So this right here. And I happen to do that at an average rate, a moderate rate. So taking all of these things into account, I have a profile, a genetic hormone profile of estrogen dominance, which can come with a whole host of things in the phenotypic way. Generally, these women have larger breasts, maybe bigger hips, more hourglass in shape. Someone who is androgen dominant may be more lean, not as curvy, smaller breasts, things like that. So that's how they can typically show up phenotypically. Okay, so getting into the step 2, our center yellow row right here. Now we're talking about estrogen metabolites. So of interest that many of you had asked about and wanted to know more about. So we have our estrogen. Now the whole point with these hormones, with toxins and other things, is they need to be broken down by the body and cleared. Hormones are one of them. Estrogen is one of them. And we have to remember that the base structure of estrogen is cholesterol. It is a fat. Okay, it has that fat base. So we need to, in order to clear it from the system, we have to go from this fat-based molecule into water soluble. And that's what the whole process is. So in the phase 1, we are breaking that down. And this is where the CYP genes come in. Remember I said cytochrome P450, part of that first phase of detoxification, because now we are breaking it down to make it more water soluble. Well, in doing that, it can actually make these molecules more inflammatory. And that is the case for the 4-hydroxyestrone and 16-alpha hydroxyestrones. These have a lot stronger estrogenetic effects, and it makes them more inflammatory. The 2-hydroxyestrone here on the left, this is more beneficial. It has a little bit softer touch when it comes to its estrogenetic effects. And actually, it needs to be methylated in order to be even more beneficial. So that's why the methylation pathway, your ability to methylate, is so important. And it works. The methylation also is very important for the 4-hydroxyestrone metabolite, because if the 4-hydroxyestrone metabolite is not methylated, it can actually create quinones that will cause DNA damage, which can lead to the problems of cancer. So
let's stop for a second.
Go ahead.
So if we look at step 2, here's what I want your takeaway to be. This 2, OH means hydroxy. 2-hydroxy, this very first one, that's your protective. You want 2-hydroxy. So especially if you're going to do the 24-hour urine or the Dutch test. The Dutch test stands for Dry Urine Test Comprehensive Hormones. You want lots of 2. 4 is the negative. That's the one that can go down, needs to be methylated to neutralize, and can make the quinones, which causes DNA damage. DNA damage causes inflammation. Inflammation can lead to cancer. So 4 is not our friend. And then 16 has a slight protective effect because it causes proliferation. Proliferation can mean more bone tissue if you're trying to rebuild. So a lot of times we'll give people estrogen if they have osteopenia, to get them back, into normal range. But 16 can also proliferate. If that 4 has gotten out of control, caused some damage, 16 can proliferate. So when you look at Dr. Lara's, you see increased toxicity risk. Why? She's slow at making 2, which is the good one. She is medium at making 4, which is the bad one, and she is slow at making the 16. So a lot like Lara, my 4 is a green. So my protective is slow, my proliferative is slow, but I go to the races with the bad one. So I have to be especially protective of not having extra estrogens, whether it's in my skincare, whether it's in my environment, whether it's in my cleaning supplies. I have to be especially protective because I know I like to slip down that slippery slope, you know? And so this is a really good one for you to understand on your genetic profile, your CYP1A1, CYP1B1, and CYP3A4. Yeah.
And I mean, honestly, when you're looking at this in more of that general sense, it actually is optimal to have all of these red, to have all of them as just a slow conversion, nice, easy. We want rates where our methylation, our other detoxification pathways can keep up and make sure that they're cleared and just keep everything working. So yeah, if you are red across the board, then you will see estrogen toxicity risk normal. It doesn't mean you're not at risk at all, because there are estrogen mimetics. There are, you know, other toxins, excuse me, that can disrupt estrogen receptors like BPAs, okay, that can cause problems. So it doesn't mean that you're totally out of the clear of having estrogen toxicity risk.
So again, BPA, in the event that you're not familiar, you've probably heard of it, but where? Teflon pans. Every time you cook with Teflon pans, BPA is released. If it's not a non-toxic pan, it's going into your food. It's a hormone endocrine disruptor. Plastic water bottles, which is why you'll notice I've got a stainless steel bottle, and Lara's got a hard bottle. Okay. Why do we not use the squishy ones? Because every time a squishy one warms up, maybe you've kept it cold, but what about the warehouse where it was stored and it was hot? Every time it warms up, those plasticizers leach out. BPA can leach out. You may notice if you've got grandkids or young kids, it'll say BPA-free camping supplies, BPA-free, silverware. Why is that important? Because that can mess and increase your estrogen toxicity, which could lead you toward inflammation, which could lead you toward cancer. And we're trying to help you. What are the things in your daily life that are under your control? Your genes aren't under your control from the aspect of what did you receive. What's under your control is the epigenetics surrounding it, your pathways, your detox pathways, as well as what you put in your environment, because those in the environment helps drive. Okay, so you may have just this neutral, but then you put all these BPAs in, and then it just slams on the accelerator and drives you down a pathway that you don't want to do, and then off that cliff. We don't want you to go off the cliff.
And please be mindful that if it's plastic, just be conscientious, because even if it says BPA free, they could be using BPS or another analog of that. It does not mean that that's safe. So honestly, I personally bring bamboo utensils with me. I travel with them, so I don't have to use plastic, or use metal or, something like this. But please make sure that you are not cooking with any plastic. If you have any, start switching them over, get rid of them. So you want to lower your exposure. So actually, I saw a question here I did want to, in the chat, Robin had asked, what if we're co-dominant? So if you are co-dominant, that means that based on all of these other pathways that testosterone can take, that you could be androgen dominant, or you could be estrogen dominant. What really determines the dominance you lean into is your diet and lifestyle. So, it's kind of like that teeter-totter that, it's so delicate that if you are exposed to a lot of different endocrine disruptors or estrogen mimetics, it can tilt you very easily one way or the other. And, so it's even more important that depending on what you're looking for or where you are, how you adjust will make that difference. So, okay, so now we have our estrogen metabolites. Again, this is the phase 1. Now we have to get into the phase 2, which is that conjugated pathway. This is that part that we have a mostly water soluble or a semi-water soluble metabolite. Now we have to make it fully water soluble. And the way that we do that is by tacking on a methyl group or a glutathione molecule, or one of the other molecules to help move that out. So that's what these are here for. This deals with your methylation pathway. This deals with your antioxidation pathway. The GSTs are your glutathione conjugation pathway, and COMT, that works with the methylation pathway because that's that end enzyme that actually tacks on the methyl group, kind of like that sticker that we were talking about the other night when Dr. Krista and I were talking about the COMT gene. So my methylation pathway is not so good. I need help with that one. I actually have the worst MTHFR, about 70 to 80% less enzymatic reaction. So I really need to support my body with B vitamins, with, things like trimethylglycine. And, I do supplement as well as making sure that I'm eating dark leafy greens and having the different, foods that provide the necessary nutrients my body needs to support my methylation pathway. So I can convert, my 2-hydroxyestrone into the more beneficial one. So I can convert the 4-hydroxyestrone in order to be cleared and not damage my DNA.
So one point that I want to bring out, don't you love this? I just think this is so fabulous. But when we look at our step 2 and we look at it, phase 1 liver metabolism, this is why we say your liver is so important. This is why if you have fatty liver disease, if you drink a little too much, maybe 1 glass is okay, but you hit 2 or 3, estrogen goes through the liver in phase 1 metabolism, which is this step 2, and becomes more toxic, okay? Then it has to go back through the liver into phase 2, that's our step 3, and become less toxic, non-toxic, and eliminated. Phase 1 and phase 2 are critical. This is why I am not a fan of regular HRT, hormone replacement that is oral. Oral estrogen, primarily people may know conjugated estrogen or Premarin. Oftentimes we can't get it because of our livers. We can't get it all the way to that phase 3. Did you see that? Okay, how fast are you converting it to water? And then do you have your methyl groups? So the SOD stands for superoxide dismutase. That's eliminating your free radicals. Your glutathione. Some of you don't have the ability to have glutathione transferase, and so you've got to use NAC. So it's like, do you know? Do you know your pathway? Do you know if you're set up? If you've got your DNA 360, you can find out. And this is why it's critical. If you're going to use hormones, it's like, I'm like Lara. I've got a really poor methyl pathway. So, and a lot of people in my family have had heart disease because they don't methylate. We build up our homocysteine. But here's the fascinating thing. People are so hung up on MTHFR, and yet we do multiple pathways, 3 and 4 pathways down the methylation. MTHR is not the only name of the game. So we've got all these others. If MTHFR is Ford, what about Chevy? What about Nissan? What about Toyota? Well, we've got, the other ones that we're working with and show you, okay, your MTHFR is fine, but your MTRR isn't, or your MTSR isn't, or those are okay, but your other is. So you can make better decisions when you have more information. So first thing, Do you do liver cleanses? Do you do intermittent fasting? Have you measured your estrogen to know, and especially estrogen metabolites? Are you going down the 2-hydroxy, 4-hydroxy, or 16? So when I had my Dutch test done, guess what I found? I found my 16 was my majority. Even though I'm a slow converter, it was pushing down there. Now I see on my profile, just like Lara has on hers, that she has an increased risk. But I was like, increased risk, smish, till I looked at the numbers and went, holy roly, I had a minor amount of protective, I had a major amount of proliferative, and I had a minor amount of bad, but I really didn't have a whole lot of good. So I did our DNA 360. I actually met the founder of the company that produced this, introduced him to Tracy. Then we took on and we brought out the DNA 360. I was the test patient. Of course, I'm fine. I have great boobs. I know how to do this. I have clearance. My test came back clinically significant. Let me tell you what a slap in the face it is to be a practitioner, to know about this, to be doing all the right things, but to come back with low-grade chronic inflammation that could lead toward breast cancer in the future. Let me tell you, if that's not a sit down and wake up call, but why was it powerful? It was powerful because it was test, don't guess. It was powerful because what I'm doing is good, but it's not good enough. It was powerful because I could empower myself to make a change before it became clinically significant further. I had a full MRI, or I had a full mammogram, clear. I had an ABUS, which is a ultrasound, clear. We repeated blood work. I brought my estrogen way down. We repeated a Dutch. I brought my 2 way up. The beautiful part of this is that this is in your control. Lifestyle, attitude, food, and supplementation is in your control. Do you feel more empowered even now, knowing, hey, I didn't know about these pathways. Hey, I didn't know I had them in my control. Hey, now I'm going to take a deeper look. I'm either going to go back and look at my test, and you can look at it yourself, and you can come back and watch this again. You can set up a call with a practitioner. You can join our upcoming She Summit that we're getting ready to do in May. You can have this information, and information is powerful because we want you to say, hey, I need my hormones done. My doctor doesn't do them. We can get them done for you. Hey, my doctor will do them, but my doctor doesn't know how to read them. My doctor doesn't believe in Dutch tests for a postmenopausal woman, only for premenopausal. Well, that's hogwash because there's lots we can find about going down the pathways. So if you need information, then go ahead and put in the chat that you want to receive a call and have one of our team reach out to you and show options so we can look at your hormone profile. We can get you a DNA 360. We can help you navigate through, okay, where do I stand? What would I do? Go ahead and put in, I'd like a call, or you can put your email in, or you can say, reach out to me, and we'll have the team reach out to you and see which option is best to get you tested, to help you have a follow-up, to help you feel empowered to move forward. And I can tell you, mamas and grandmas, if you know what your profile looks like, you have a tendency, not exactly, but you have a tendency to know what your daughters and granddaughters could be exposed to. And then, well, how about this tribe all working together, going, hey, what are we going to do for Christmas that's non-inflammatory? Hey, maybe we give each other non-toxic cookware. Hey, maybe we get everybody, mugs, and it says Nielsen family on them, but they're all non-toxic. There are ways to make this fun, but also healthy, because there is nothing as powerful as being healthy is.
Thank you so much for sharing that.
Yeah.
It is so empowering and so wonderful. And it is fun to learn. I mean, at least I find it fun to learn. I consider myself an academic masochist because I find this so fascinating. And I do know that one of our, I saw in the chat about talking a little bit more into step 3. So again, this pink row down at the very bottom, this really deals with that phase 2 detoxification system, really putting, the methyl groups or a glutathione for that final clearance, turning these estrogen metabolites into water soluble molecules, so that way it can be excreted from the body, through urine. This is how the Dutch test actually reads that. So for example, like I said, my methylation pathway is not great. The guy, my comp putting that sticker, that methyl group on, works at a moderate rate. There are ways that I can increase it or decrease it as the case may be, whatever, I happen to find on my tests, which actually there was another question I saw about, does HRT affect your Dutch test? Most definitely.
Mm-hmm.
Absolutely. Actually, that was something that I did because I'm postmenopausal. I started on bioidentical hormone replacement therapy. And again, the tests don't guess. No, I wanted to know, how is this working for me? How is this therapy showing up? And absolutely, I, took a look, of course, also working through supporting my detox pathways, the way that I ate, trying to eliminate or lower my toxic exposure. So when I did my Dutch test, obviously my estrogen levels were still a bit on the low side because I had just recently, started on that. And it takes a little bit of time trying to find the right amounts that you should be taking, how your body is working through. But my ability for the percentages, and there is an optimal percentage for each of these, for the metabolites, actually was fantastic. It did not reflect more of the 4-hydroxyestrone because of the foods that I was eating, the cruciferous vegetables and, sulforaphane and different aspects like that, that helped to support my methylation pathway to make sure that I had the correct percentage for each of these metabolites. The only thing is that I did notice that there were some things that I needed to support a little bit more. I wasn't actually methylating my 2-hydroxy quite as well. I was, but not quite as well. And, there were some other levels off. So it gave me and my practitioner information so that way I could further tweak, going, oh, I need a little bit of testosterone here. Oh, well, maybe we'll shift and add in, maybe a little B6, or, maybe putting in some calcium D-glucarate, or, and I'm getting ready to test again to say, okay, with these changes that I made, let's see how we're doing now. So very important. So in this, take a look. You will find the information for these particular detox pathways in the anti-inflammatory reports, and then sub-reports. They actually break it down. Like it will say glucuronidation. It'll say methylation, antioxidation. That's where you'll find the SOD2 and GPX. You'll have your vitamin D. Yes, vitamin D is very important for detoxification and very much impacts hormone balance, hormone health, brain, gut, immune system, everything.
So there was a question about finding this chart, and I misspoke. As a clinician, I see it all the time. I misspoke. They're put, like Lara just said, they're put in different parts of your patient reports. That's pretty intensive. I like showing it just because, and she says she does too when she works with an individual client. She lets them, have their chart. You will not find that in the regular patient reports, but I want to remind you that we have our DNA Co app. That's a free app. And with the DNA Co, you can get the app. Your report will be, you can upload it there, and you can find all sorts of data about stuff here. And if you want to do some research on that, there's tons of information in there that you can find your different things. But Lara, we've got over 20 questions. I think we
I
better
know,
start
I
hitting
wanted
them.
to jump into the questions. And
Okay.
actually, I noticed that this first one, Alicia, you had asked, does this apply to triple negative breast cancer? Unfortunately, I don't remember exactly what was said that you are referring to. Does this apply? Does what apply?
Well, she did it real early. I think we were talking about percentages and BRCA genes.
Okay, if Alicia, if you can throw that in the chat real quick, just so we have clarity on the question, so we can give you an appropriate answer, that would be fantastic. As she is doing that, let's jump to the next one. So Joseph, he says, Dr. Dennis Burkitt and Dr. Albert Schweitzer noted in their years in Africa that cancer and heart disease were essentially absent, and noted that their diets were very high in fiber. Comment on this as a mode of breast cancer prevention and the amount of fiber you recommend.
So I think they're absolutely right. There's more and more information about not only parasitic infections, as well as fiber contents related to multiple kinds of cancer. Tonight, we're talking about breast cancer. The thing about fiber is fiber can absorb a lot of toxicity. And it's not a single fiber. It's not just like Metamucil. So you've got to have multiple different fibers. I actually take a fiber supplement that has 5 different types of fiber in it. So that works from the top of my system to the bottom of my system and rotates all through the different ways. In addition to that, when people are looking at heart disease, there is one drug, cholesterol, that actually absorbs the different cholesterol. Cholesterol is the mother of all hormones, breaks down to pregnenolone, then breaks down into all these other hormones. It absorbs that and pulls it out. So I totally think that you've got to have at least 30 to 50 grams a day, which is woefully lack in a processed food diet or a SAD diet. But yes, I agree that that is a phenomenal thing. And if you look at having flaxseed with quark or cottage cheese, that is also recommended in a lot of anti-cancer treatments. And that flaxseed is the flax fiber and seeds, so that you're getting your omegas in there as well as the fiber. So I totally agree with what they're saying. Fiber, fiber, fiber.
Yeah, and I'll tell you, having that backed up by science, there was actually a 2016 meta-analysis of 20 observational studies that found that with each 10 grams per day increase in fiber intake was associated with a 7% reduction in breast cancer risk. Mm-hmm. So absolutely.
Janice was saying, what exactly does codominant mean? We kind of answered this a little bit. What effect would the profile have on estrogen toxicity or conditions that lead to breast cancer? Do you want to just kind of redo codominance for a moment?
Okay, so when we're talking about codominant profile, that means that you could lean into either being androgen dominant or estrogen dominant. And really, when its influence on breast cancer, that would actually, there would be more information that I would want to look at in the DNA profile to determine more to that, because it's more than just your genetic hormone profile. I'm also would really want to look at that second and third row. I'd want to know, are you more genetically inclined to have an increased rate of making any one of those metabolites, especially the 4 and 16 alpha hydroxyestrones, because those are a lot more inflammatory, and therefore increased inflammation, especially when you're talking about having it happen in fat tissue, because it will get stored in fat tissue. Toxins, fat soluble toxins, get stored in fat tissue. Where do women have the most concentrated fat tissue? Their breasts. Mm-hmm. So, you know, again, that, and I'd want to see like detox pathways, how well are you detoxing, and then be asking questions, but strictly on the genetics and the codominance. To me, that's That's kind of like looking at the Mona Lisa through a straw. I see a beautiful picture of you, but not enough of it to really get the full picture, you know, to give a more definitive answer on that.
So April asked, is sulforaphane more helpful in a slow comp type versus a val fast type?
Okay, so for that, hold on, I have to do a quick think
I was going
here.
to say, I don't know what the val is.
It's a valine.
Or-
It's just the amino acid, the protein. So, okay, sulforaphane, first of all, it's the slow comp genotype is the met, which is methionine, compared to valine is the fast comp, okay, for fast clearance. So sulforaphane, it can be helpful for individuals with slow as compared to those with fast. So if you're saying about, hey, if you've got a fast type, yeah, sulforaphane is, I mean, it can be helpful, but maybe not as much so. Really, when we're talking about the COMT, remember this is catecholomethyltransferase gene. So it's breaking down those catechols, so the estrogens. And for the fast clearance, is it something that you want to, wait, helpful in slow types? Oh, versus fast types. I'm sorry, I read that fast. So yes, the sulforaphane does help with the slow type.
Yeah, I would say so.
I was kind of reading that fast and then it was jumping over into my brain of trying to think. So yes, sulforaphane can help. Sulforaphane, lean into foods. Broccoli sprouts is great for this. Cruciferous vegetables can get you those.
And that's also where people end up when you're trying to move the sulforaphanes. A lot of people end up using DIM. I don't like to use DIM indiscriminately. You've got to make sure that you're using DIM if you know your estrogen levels are high, if you know that they're going down the wrong pathway, because it's going to clear the estrogen out. If you've got good estrogen pathways and good balances, why do you want to clear it out? That's not what we want to do. I don't recommend DIM unless we've seen a 24-hour urine and we've seen the metabolites, or we've seen a Dutch and we know which pathways we're going down so that we can drive that bus more clearly. So I don't know if it's Gian or Gon. DIM definitely helps with estrogen dominance, but you've got to make sure that you need it first and you don't just take it indiscriminately. Yeah. How about
I love, yeah, this next question, April, you know, about how protective is that the fast comp or the okay, that the val is fast. Okay, so let's just get that clear. How protective is a fast comp if someone has a really bad methylation pathway in general, genetically? And that is such a great nuanced question.
Because that's nuanced.
Yeah, because you know, when you consider a fast comp that might look protective at first glance due to that faster breakdown of estrogens, catecholamines, etc., but it really depends heavily on the full methylation pathway.
Yeah.
And why this is important is because even if you have a fast comp, it's only as fast as your methylation allows. It's like having a relay race. Okay, so each of the genes, each of the players, it's passing the baton, the baton being the methyl group. Okay, Compt is your final runner. He's the last leg. Okay, so he can't really do his job. He can't, you know, run it in until the other players pass him that baton. And if they're slow doing that, if they're not very good, if they keep dropping it, then Compt, no matter how fast he is, it's gonna be a problem. So yeah, if you do have a sluggish methylation cycle due to genetics, then the Compt can't function quite as optimally, even if itself is fast. But that's where the support of the methylation pathway can help. And these are like with your B vitamins, trimethylglycine, inositol, things like that, that can help them, those runners, making sure that they're not dropping the baton, that they are faster. So that way comp can be awesome and fast and do the job that they're doing. So great question. All right, so Joseph has a question. Can you comment on commonly used drugs that interfere with estrogen detoxification and or raise estrogen levels?
I'm
So
trying to
Dr.
get a
Stephanie,
little, yeah, I was gonna say, I'm trying to get a little bit more information before we answer that. Let's answer like a couple of the other ones,
sure.
and I'll come back to this, Joseph. I wanna look at a couple things quickly before I just shoot off an answer for that.
Okay, so his next question is, how does the microbiome affect estrogen detoxification? I love this because I love the microbiome. It's so important. Your microbiome plays a huge, often overlooked role in estrogen detoxification. And, this really is mediated by a specialized subset of gut bacteria called the estrobolome. Okay, and I know that's a funny word, estrobolome. And it really refers to the collection of gut bacteria genes that metabolize the estrogens, that regulate that estrogen recirculation through the enterohepatic system. So we're talking about like gut and hepatic, meaning liver. So, working through there. So basically these are microbes that produce enzymes. Really a lot of it, the main enzyme is the beta glucuronidase. And that can actually reactivate estrogen in the gut and send it back into circulation. And that is that estrogen reabsorption, which is not always a good thing, just to let you know. So really, we've already talked about how estrogen detoxification normally works, in the phase 1, the phase 2 detoxification, that elimination, generally through bile, the intestines, and through their urine. And having a healthy microbiome, those microbes that keep conjugated estrogens bound for excretion instead of being, recycled. So when you have dysbiosis, okay, that imbalance of the gut flora, that can lead to higher levels of beta glucuronidase, and that uncouples the estrogen. Instead of it being bound to be eliminated, it uncouples it, and now it's free and circulating again. And it sends it back into the bloodstream. And like I said, that's not such a great thing. So this is how the gut affects, there are more nuances and stuff, but generally that's how the microbiome can affect estrogen detoxification. So,
So, so Joseph, as far as commonly used drugs to interfere with detoxification or raise estrogen levels, literally, I put it in because as a pharmacist, I'm going, well, I'm not thinking of anything off the top of my head. And so I was like, oh, yikes, there must be something. It doesn't appear that an average drug will interfere. Obviously, we have all of the aromatase inhibitors, which slow the breakdown of testosterone to estrogen. And the reason that's important is the tamoxifen or the anastrozole or some of the other things that we use when we're not wanting to increase estrogen and we're not wanting to have those breakdowns. Those obviously manipulate levels. I think the more common thing that we see are things that mimic estrogen, phytoestrogens, where it ends up raising, something sits in an estrogenic receptor. Because remember, we have estrogenic receptors not just in our breasts, we have them in our brain, we have them in our gut, we have them all over our body. And it happens to be that we raise that estrogen impact, that estrogen effect, but maybe we don't actually have the actual estrogen, but we have things that are mimicking that. So I can't find a day-to-day drug that I'm aware of, like a blood pressure medicine or like a diabetes medicine that causes any problems with that. The biggest thing is liver's got to be functioning well. You've got to have that healthy liver if you want to detox and you want to get it down to those healthy levels, and then keep away from those things that are going to interfere and sit in the estrogen receptors.
So I'm going to grab Jennifer's question because she says, what report shows which of the CYP1A1, CYP1B1, and CYP3A4 are the slow or fast variants? And in order for me to do this, honestly, again, I love visuals. I'm going to share screen really quick and show you. Okay, can everyone see that?
Put a
Can
1
you-
in if you can see it. If you can see the screen, Dr. Lara's screen.
It's there, Dr. Nielsen.
Beautiful.
There we go.
Perfect.
Good.
Okay, so when you are looking at your reports, go over to hormones, fitness, and body type. You're going to click on that. And then you're going to go to system summary. And it's within the system summary that gives you all this wonderful information. It gives you your gene results, hormone profile, general health outcomes, genetically based. This does not look at lifestyle or anything like that. This is strictly just on genetics. And this is where that descriptor of each particular gene, the genotype, and then the description that tells you, oh, hey, your CYP17A1 is fast. Oh, hey, your aromatization, the CYP19A1, that's medium. That's where you will find it. There we go. Okay. So next, let's see. So Melanie, she's saying, I had ER positive, PR negative BC breast cancer. How important is progesterone in keeping estrogen in line? Did you want to answer that?
Well,
Or is this?
Yeah, everything, as you are alluding to, Melanie, is in balance. When you have estrogen positive, progesterone negative, progesterone is very important. They all balance and work against each other. How long ago, maybe you can put in the chat, did you have this, and what did your primary care or your oncologist say to you about doing it? Because what we see, and of course we don't deal directly with breast cancer, we might get breast cancer survivors, and we deal with people who are perimenopausal. Maybe they want to look at balancing their hormones and understanding. So at this point, what I would say is I find it very important. 5 years ago, I had breast cancer. Oncologists don't normally test hormones. I would, for your sake, get a Dutch test. Again, Dutch is a dry urine comprehensive. That's going to show you the pathways going down. It's also going to show you your levels. We can order a Dutch for you, and then I'm one of the providers that does follow-ups on those. It is so well worth the investment. Dutches are not inexpensive. They're about $500 or $600, but you only have to do them every 1 to 5 years, and especially post breast cancer. It is really critical to look, hey, which pathways am I going down? What does my DNA say versus what am I actually doing? And how do I stay out of that proliferation? How do I keep in that green zone, which is that 2-hydroxy? I think that may be a real win. And I see you say you did have a Dutch test. I would look at that Dutch test that you had 5 years ago versus what you're doing now. And then there's lots of lifestyle supplementation that we can look at to make sure that you're keeping that balance in what we see. So, yeah.
Okay, so we have, Melanie also is asking, on the stoplights, I thought red would mean slow, not good, but it was stated that you want all red in step 2. I don't understand why slower is better. Okay, so we need to uncouple the speed of which and whether it's good or bad. because it's a matter of it just is. And where does that work into the overall pattern, the overall, let's go back to sports players. Okay. So say you have a basketball team. Well, you have some that are stronger in some areas, weaker in others, that have, good stats and some and not so good. But the point being is that one player doesn't play the game. It's all of them playing together. And it's how does that entire team play? So first of all, uncouple the speed and whether it's good or bad. And when I was saying that all red in that center row, it's because you don't want too many metabolites. Remember how Dr. Stephanie was talking about that during the process, during the phase 1 process in the liver, as these molecules are being broken down, they can become even more reactive. They can become even more inflammatory. And you're probably wondering, why would the body do that? Well, it's for 2 reasons. One, it has to break down these molecules to make them more water soluble in order to get rid of them. And it's just something that happens. It makes them, more so. Not always, but oftentimes it does. But it also is acting like a neon sign saying, hey, come take care of this. Come, let's make this molecule a priority so that way we can move it along the detoxification pathway and get it out of the body. So the thing is we don't want this to happen too fast, making these metabolites, because it can overload the liver. The liver is already working so hard dealing with regular toxins and regular metabolites, just the trash that our cells are making. It's got a job to do, and it's constantly working. And then you throw on all of these other toxins we're exposed to in our air, our water, our food, that we are just inundated with. It's like, let's not overload it. So having a slower, steady, and allowing the liver to do its job, that's why I had said having a slow rate is actually optimal, giving the liver a chance to do its job and not overload it.
And this actually goes right into Joseph's question. If the Dutch test has all 3 estrogens and their metabolites are pretty low, but your ratios show a higher percentage of the 4-hydroxy metabolite, is it still significant even though it's low? The answer is yes, yes, and yes. It's yes, because it's the same. I want you to take a plate. You've got protein, you've got fat, and you've got carbohydrates. If your plate is very small and you only have a few things on it, but 90% of what you've got is carbohydrate, is that a balance? And are you going to get healthy eating simply carbohydrates? No, you're not, especially processed carbs. So that 4-hydroxy goes down and can cause the quinones, and the quinones cause the DNA damage. So even if you have low amounts of estrogen, when they're out of balance, you risk the rate of more damage to your DNA and more inflammation. So that's why we say to people, it's okay to do the Dutch test even postmenopausally, because we want to look at those ratios. We want to see what you're converting. A lot of women say, oh, all of a sudden I've got a beard. All of a sudden I've got, my hair's falling out. Well, if we know that it's pushing down, we can help them, food, supplementation, or whatever, to push it back the other direction. And that gives us that. And Dr. Lara, I hate to do this to you. I had a hard stop at 6. My phone is blowing up. I have a client. I got to go. Thank you guys for coming.
Bye.
Thanks for staying. She'll take you out, and we appreciate you all. Have a great night.
Thank you so much, Dr.
Bye-bye.
Stephanie. We love you.
I love you too. Bye.
So I'm going to answer this last question by Rhonda, and she's saying that the graph was awesome. Thank you. I'm so glad that you liked it and that you took away some information from it. And she's asking if I'm able to say the gene version in the top circle for each gene on the graph. Now, I'm suspecting you're saying the green or the fast version of some of those. So what I'll do very quickly is the genes that are related to those particular stop signs, that determine what your hormone profile is. So the first one we talked about was the conversion of progesterone to testosterone. That's your CYP17A1 gene. And if you have a G allele, that means that you're going to have an increased rate of that conversion. An A allele is a slower conversion. The next one was the UGT genes and the CYP3A4. So really the UGT gene, so this was UGT2B as in Bob 17 and UGT2B as in Bob 15. So the one that ends in 17, that's a copy number variant. So it's actually the conglomeration of those 3 gene types. And then, we look at that. So depending on if you have 0, 1, or 2, obviously 0, you won't be clearing very well. This is your glucuronidation pathway. A 1, moderate speed, 2, fast. But then we also have to look at the one that ends in B15. A T allele is fast, G allele is slow. Obviously, if you have a GT, well, then it's a moderate rate. And then the CYP3A4, which actually works into how quickly we metabolize our estrogen into the 16-alpha hydroxy estrone metabolite. If you have an A, that would be a slow rate. A G would be a fast rate. So we take those 3 and then determine the speed of clearance of your androgens. The next one was the AR, your androgen receptor. So the androgen receptor, how well you are binding your testosterone to the receptor. The C allele is an increased rate. The T allele is a decreased rate. Then we have the SRD5A2. It's the conversion of testosterone into dihydrotestosterone. A G allele is fast, a C allele is slow. And our last one was the conversion of testosterone into estrogen, the CYP19A1 gene. A T allele is a fast conversion. The C allele is a slow conversion. And I know I said that kind of fast, but thankfully these are recorded and you can always go back and listen, and then you can play it, pause, check yours, play it, pause, check it again. So, but I do want to be respectful of everyone's time. And thank you so much, everyone, for staying on, for learning, for asking your questions, for being such a wonderful interactive audience. We appreciate you. So to that, I say thank you and have a wonderful night, everyone. Take care.
Thank you so much, Dr. Barden. Thank you, everybody. Have a good night.